Hello and welcome back to the GCP-Mindset channel. Today we'll talk about biometry in clinical trials.
Welcome to the GCP-Mindset channel and all topics on clinical research.
Let us delve into understanding biometry in clinical research and why it is so important. Biometry, also known as biostatistics, plays a pivotal role in the design, analysis, and interpretation of clinical trials. It ensures that studies are scientifically sound, ethically conducted, and statistically valid. This video distills some of the most practical and critical aspects of biometry in clinical research, providing a roadmap for researchers, clinical trial associates, and statisticians. Let us start with what biometry is. Biometry refers to the application of statistical methods to biological and clinical research. It encompasses study design, which involves planning how to collect and analyze data collection, where methods are structured to gather accurate and reliable information, data analysis, applying statistical models to derive meaningful insights, and result interpretation, ensuring conclusions are valid and scientifically robust. Now, let us explore the core principles of biometry in clinical trials. Clinical trials rely on three fundamental principles. First is randomization, where subjects are randomly assigned to treatment groups to reduce bias. Second is replication, which ensures trials include sufficient subjects to guarantee reliability and reproducibility of results. Third is local control, which involves minimizing variability and controlling influencing factors. Every clinical trial begins with a clear primary objective and well-defined hypotheses. The null hypothesis, or H0, assumes no effect or difference, while the alternative hypothesis, or HA, suggests a measurable effect or difference. The choice of study design, whether single-arm, multi-arm, crossover, or sequential, depends on the research question, ethical considerations, and logistical constraints. Randomization and blinding, whether single, double, or open label, are critical for minimizing bias and ensuring credible results. Let us talk about statistical tests and sample size calculation. Statistical tests are selected based on the type of endpoints being measured and the study design. Continuous variables, such as blood pressure, often require t-tests if the data are normally distributed, or regression analyses for more complex relationships. Binary outcomes, such as cured, not cured, may use chi-square tests for simple comparisons or logistic regression to account for additional covariates. Survival data, including time-to-event analysis, are commonly analyzed with Kaplan-Meier curves for descriptive purposes and Cox regression for modeling relationships. Sample size calculation is a cornerstone of trial planning. It accounts for the alpha error, type 1 error, the probability of falsely rejecting the null hypothesis, typically set at 5%. Beta error, type 2 error, the probability of failing to detect a true effect, is often set at 20%, corresponding to a study power of 80%. Effect size, the minimum detectable difference, and standard deviation, a measure of variability in the data, are crucial inputs. For binary outcomes, the prevalence of the event in the population is also considered. To address bias and ensure validity, robust randomization methods and allocation concealment are critical. Double-blind strategies protect investigators and participants from knowing treatment assignments, reducing bias. Statistical adjustments, such as multivariable regression or propensity score methods, help account for confounding variables and strengthen causal inferences. Now, let us compare drug and medical device studies. Drug studies typically require larger sample sizes than medical device studies due to the systemic effects of drugs and the need to monitor multiple endpoints. In contrast, the localized effects of medical devices often lead to fewer side effects and simpler endpoints, allowing smaller sample sizes. Here are some practical takeaways. Early involvement of biostatisticians ensures robust study design and analysis plans. Choosing the right statistical test and sample size is vital for reliable results. Understanding the differences between drug and device trials can optimize resource allocation. Finally, let us conclude with why biometry matters. Biometry provides the backbone for designing, executing, and interpreting clinical trials. Mastering its principles and applications enhances the credibility and success of clinical research. Whether dealing with drugs or medical devices, integrating biometry at every stage is indispensable. By grasping these core concepts, clinical researchers and associates can ensure their studies are well-structured and their results impactful. Thank you for watching our video, and we hope that you found it informative. Don't forget to like and subscribe to our channel for more content on clinical research. We look forward to see you next time.